for the, I'm gonna click to agree to that real quick, as well as to the people who are interested in looking at it for biofuel feedstock as well. So more specifically, I want to talk about special defense of aspen, which are these compounds called phenolic glycosides. They're important in, they're a secondary metabolite, and they're important in herbivore defense in Salicaceae, the family that Populus tremuloides is part of. And they're particularly abundant in the foliar tissue of these plants, the leaves of the trees. Sometimes it can be up to 30% of the plant dry weight, which is a lot. They've also been shown in the past to reduce feeding and survivorship, growth, and reproduction in a number of lepidopteran pests of aspen. So they're an important defense against herbivores for the trees. Some previous work in the lab has looked at phenolic glycoside concentration in aspen genets, which have shown a lot of variation. So you can see on the left, if you look at the red box, those are two of the phenolic glycoside compounds that are most abundant, tremulacin and salicortin, and each bar represents a genet. And there's a lot of variation in how much of the percent dry weight of the foliar tissue that was measured for this is composed of those compounds. So it can be really low, close to almost zero, and then it can be up to 8% for this example. And then the other thing that was noticed too is that there is a negative relationship between growth of these trees and how much of these PGs that they have in their tissues. So that's kind of some background information. I'm going to talk about the WISAS garden now. So we call it WISAS for short. The long name is the Wisconsin Aspen Association Mapping Garden. It was established in 2010. It's at the Arlington Agricultural Research Station, if any of you have ever been there. And here's a nice drone view that Chris Cole from our lab took on the right there, showing all of our trees. There's 1,824 trees in this garden. It's kind of crazy being there because you're like walking through like a forest in the middle of all of this farmland, especially now that the trees are so big. So the way that the garden is designed is it has 4 replicate blocks of a total of 515 genets that were collected from a north-south transect in Wisconsin. And from 2014 to about 2017, we've evaluated a number of traits that are important for growth and defense of these trees, including tree height, diameter, breast height, leaf morphology, bud break, so when the trees leaf out in the spring, and then obviously phenolic glycoside, and then condensed tannin concentrations. So that's the phenotypic data. We also have had all 515 genets exome sequenced via sequence capture genotyping, which basically what that means is you take some probes that you've designed to go to the portions of the genome, in our case genes, that you're interested in sequencing, and then you sequence those. It's pretty effective and both in efficiency and then cost way to get a lot of individuals sequenced. So we did that, and then we took our sequencing data and we put it through a filtering process, a pipeline that we designed. And once we did that, we had 291,069 SNPs in 19,752 genes that we could use for our genome-wide association analysis. And I will talk about that shortly in more detail, but first I'm going to give you a little bit of background on the phenotypic trends for WISAS. So here are some graphs that are kind of showing the variation in growth and defense traits at WISAS. So you've got size on the top, and then PGs and CTs, so phenolic glycosides and condensed tannins, which are 2 defense compounds for aspen, below that. And their concentrations or volume in cubic centimeters squared, or cubic centimeters, sorry, are on the y-axis, and then on the x-axis, each black bar represents a genet. And remember, there's about 500 genets in this garden. So you can see from looking at this that there's a lot of variation in how much how much of the foliar tissue has PG compounds in it, anywhere from 2 or 3% to 20%. And also the size varies a lot too for the trees. So that's kind of showing the variation there again. And this is something that you want to see in an association mapping garden that you want to use for GWA, because if there isn't any variation, it's going to be kind of hard to do with GWA. And then also we wanted to see if you were seeing the same kind of growth defense trade-off at WISAS like had been seen at previous common garden experiments the lab had done. And we have some recently published work that does show a negative relationship between growth and defense, even when you do account for age, because this actually looked at data from the age of 4 to 8 for the trees. So that's still there. And just to kind of summarize what I've talked about so far, with phenolic glycosides, we know that they strongly mediate plant-herbivore interactions and that they're negatively correlated with growth, and that they're likely highly genetically controlled. But there's not very much known about the genetic architecture of PG variation or how that's related to growth. Very few genes have even been identified that are directly involved with phenolic glycoside biosynthesis. There are a few studies that have looked at it. So that's kind of where we're at. And so what we wanted to try to do with our work is we wanted to identify candidate genes that were associated with growth and defense traits using genome-wide association analyses, which, in case anybody's not entirely sure what that is, essentially it's like a regression analysis where you take the genetic variants, in our case SNPs, as fixed factors that are individually fitted for an association with the phenotypic traits that you measured. For example, PG concentrations or size, tree size. And then you use that to identify SNPs, in our case, that are correlated with that phenotypic variation. And you do this one marker at a time. So that's why we call it I'm calling it single marker GWA, and I'll explain why that's going to matter later, because I'm going to talk about another type of GWA that looks at all the markers at the same time. So within the single marker GWA category, we ran both univariate and multivariate. And really what that means is with univariate, we're just looking at one trait and seeing how the variation in that trait is explained by our SNP data set. With multivariate, what you can do is you can create like a combination of traits that are related either functionally or because they're genetically correlated, for example. And the usefulness of that is that it can sometimes improve the power of your GWA, especially when you have genes that are likely to act on more than one trait through a functional or genetic correlation. So like an example of that would be with the 4 PG compounds that we look at the most in our trees, tremulacin, salicortin, salicin, and tremuloidin. Those all have like a functional relationship to each other. So you could put those together and then run that against your marker set and see if there's any correlation to that suite of traits instead of looking at them individually. And then we also did a differential expression analysis, which I'm going to explain more about how that was set up later. And then we wanted to look at what the genetic architecture of these traits were, and we're going to do that through a multi-marker GWA, which is essentially where you take your entire marker set and see how much variation in your trait is explained by all of your markers at one time. And I'll kind of explain how that works more later. But that's kind of the outline of what we wanted to do and how we were going to do it. So I'm going to kind of jump right into our single marker GWA analyses, the results for that. So this is for univariate, so just one trait. The first thing you can notice is that the number of candidate genes column, there's not very many. So For defense, we only found 2 genes that had significant SNPs in them. And it was only plant hormones that were involved in that, jasmonic acid and then jasmonate isoleucine, which are involved in regulating defense. So those hits, the first one is actually somewhat interesting because it has been linked to being involved in increasing transcription of defense genes in switchgrass. So that makes sense from a biological perspective, that it would be linked to a phytohormone involved in defense. And then with our growth traits, we only found 3. They were genes that are involved in sugar transportation and then also morphogenesis, which makes sense for growth. Damage, one of the things that came up was this F-box caltropete, and that is involved in phenylpropanoid pathway regulation, which is the pathway that includes making phenolic glycosides and condensed tannins, which are both defense compounds. And then also another one, this metalloprotease WSS1-like, which is involved in repairing damaged DNA that's been damaged specifically by reactive oxygen species and has been linked to response to pathogen attack, essentially. And then with phenology, a lot of the things that we found for that were related to organelle biogenesis, which would include chloroplasts, transportation of hormones and other compounds across the thylakoid membrane, which is part of the chloroplast that does the photosynthesizing. And then also some regulation of ABA and JA pathways, abscisic acid and jasmonic acid pathways that are involved in phenology and senescence, and also abiotic stress response. So even though we didn't find very much, most of the results we found were biologically relevant. There were some of these genes that we didn't really have much known function on, like this long chain alcohol oxidase FAO4A1 under defense. There's not really much known about that. But the other thing to note is that this is actually not that different from what other GWAS have found that have been done in similar species, like P. tremula, which is a close relative of tremuloides in Europe, and Trichocarpa, which is the model aspen species. So we're not finding things that are too different from what other people have been finding. One thing I did want to point out that was really cool that I saw was that we actually had sex information for all the trees at our garden. And so I went ahead and ran a case control GWA for sex just to see like what we would find. And it was pretty cool. There were about 19 candidate genes that were identified for sex. And the one that was the most interesting and also the most, it had like the lowest odds ratio, was this 2-component response regulator AR17-like gene. And that one's really cool because it's already been implicated in sex determination in P. balsamifera, which is another Populus species, and also Populus tremula, which is closely related to ours. The other cool thing, I don't have this information here, is that most of the genes that were associated with sex all kind of clustered on chromosome 19. Which is where they think that this gene, well, definitely where this gene is, but then also where they think most of the sex control for a lot of Populus species is on that chromosome. So it was kind of reassuring to see those results for sex because we know a little bit more about where some of the genes that control it are located already. So I'm going to talk a little bit about the multivariate GWAS that we did. This also we did not get too many results. The biggest thing that we got out of it is for the defense category of traits. We got 6 candidate genes from that, and 5 out of those 6 were specifically related to the multivariate trait suite that included the 4 phenolic glycoside compounds. So that was really neat because we didn't get any hits for that at all in our univariate GWA. And it's one of the things that we're trying to generate more candidate genes for because there's so little known about it and it's so important to the tree. So those genes were, one of them was related to cell wall sensing, which could be important in terms of helping pass information about external stimuli, say a herbivore trying to eat your leaf. And then also there were 3 other genes that were related to defense regulation. Growth was another one where we got 1 new candidate gene that's involved in circadian clock regulation, but it hasn't been studied in plants. That's partially why it's got kind of an interesting name too, because it's only really been studied in humans and some mammals at this point. So we don't really know what role that might play in plants at this point. Phenology was the only one that didn't have results that I thought were particularly useful, but it's not that surprising. The combination of traits that were used for phenology were based off of genetic correlations, and one of the other traits that was in that trait suite was percent disease tissue. And that's why I think the F-box Kelch repeat has shown up again here. And if you look at the loadings for this multivariate analysis, which is basically like how much of the variation is attributable to one trait over another, it's the highest for disease for that particular SNP that was in that gene. So once again, this number that we found is somewhat similar to another study that was multivariate GWAS study that was done in Trichocarpa, where you found some stuff, but not a whole lot. So I want to kind of go back a step and look at broadsense heritabilities that we calculated for our traits at WSASP. And these are kind of divided by functional category, defense, growth, phenology, and then other includes sex, and then also are kind of our damaged traits, disease, or like percent diseased tissue, percent tissue that had herbivory, and then both of those combined is the total biotic damage to the leaf tissue. So one thing to notice is sex is kind of like a pseudo control in this. It's something that you would expect to essentially have like complete heritability, which it does exhibit that at 1. Because it's completely genetically controlled. Another one in here that's actually pretty high as well as bud break, which we know that's under a lot of pretty high genetic control as well. So that was not surprising to see the broadsense heritability be so high for that. It is worth noting that when you calculate broadsense heritabilities, it's not really telling you like how a trait is genetically determined exactly. What it's really telling you is how much environment is accounting for the variation in your trait in the population you're measuring as compared to the variation that's being accounted for by the genet, which is kind of like your holistic like genome for an individual. So for our growth traits, most of the broadsense heritabilities were pretty low, which isn't too surprising because environment plays a huge role in how plants grow. But then with a lot of our defense traits, including phenolic glycosides, we had fairly high heritabilities. So, you know, when you're doing something like this and you see these, you kind of get a little bit excited in the sense of like, okay, well, we might actually be able to find some interesting things with the GWAS. So then the question kind of becomes like, well, why are you not really finding very much? Well, this has been discussed for, I think, a couple of decades, at least at this point now, ever since GWAS analyses became pretty common. This idea of missing heritability, where like you're working on a population that you're doing genome-wide association analyses on, and you see that like some of the traits are pretty highly heritable, but then when you go to do the GWAS, you just don't really find much to account for that. And that's something that happened with ours. Like we didn't find that many genes, but also a lot of the genes we found didn't have very high effect sizes, you know, maybe like a portion of a percent for most of them. Bud break was the only one, that one and sex were the only ones that had like higher effect alleles. And even those were somewhere between 1% and like 5%. So, so you're not getting a lot explained. And there's several reasons for this. A lot of it has to do with kind of what this chart is outlining here. You know, what the frequency is for the alleles, and then also what the effect is on the trait. Like, does it have a low effect or a high effect on the variation in the trait? And so a lot of what gets found in most GWAS studies is kind of on this right side here, where the allele frequency for the variant is common, and you can have like lower effect if it is a more common allele, but also this high like effect area. So same here, like if it's a little bit more rare but it has a high effect, you're more likely to find it as long as your sample size is large enough. So what happens with, say, this little corner here, if it's more rare and has a low effect, or even kind of in the middle here, where you've got low frequency variants that have kind of more of an intermediate effect, is the significance tests that you have to do for GWA are pretty stringent, and that's partially because you're doing so much testing. Like I mentioned before, when you're doing a single marker GWA, you're testing each marker against your phenotypic data individually. So if you recall from earlier, we have almost 300,000 SNPs. So that means I'm doing basically 300,000 tests. You have to account for that multiple testing. And so when you do that, what happens is a lot of these low to moderate effect variants, especially the ones that aren't as common allele frequency wise, are going to get kicked out. They're not going to be found to be significant. And then also, it can be harder to detect really rare variants, like in this corner here, if you don't have a large sample size, because your sample size needs to be large enough for you to actually see those variants. And we have a decent size for our garden. We have about 500 individuals that we use in our GWA, but that's really not enough for actual rare variants. You need at least 1,000, if not like 10,000. So that's also another place where you could be losing information. And then there's also what the trait architecture is. And the perfect example for this that gets used all the time is human height. So human height has really high heritability. It's like 80%. And it was something that, sorry about that. Like struggling to figure out where my mouse went. So when they did initial like single marker traditional GWA to look for variants that were involved in controlling human height, they only found like 5% of that, like 80% of variation explained. So, you know, the question was, okay, well, if it's highly heritable, like, where is that going? Why are we not finding much with this? And one of the things that they did to investigate that initially was to essentially develop a method where they could look at, okay, what does our entire marker set like contribute in terms of explaining variation in this trait, human height, that we care about? And that's what a multi-marker GWA is. And that did explain a lot more. So I think they got up to about 40%. So there's still other things that are probably contributing to this missing heritability. And there's a huge discussion about this going on, has been going on for a while, still going on right now. And I'm not going to get into all of it right now. But first, what I want to go to is what we decided to do, which was try this multi-marker GWA approach to look at the genetic architecture of our traits. It hasn't really been used in outcrossing plants like ours. It's been used in crop species a little bit, corn and rice mainly. And there's only one other tree species that I know that has done a study like this at this point. So it hasn't really been used much. Most of these methods use like a lasso or ridge regression. That basically takes the effect of multiple markers on a phenotype. You evaluate it like at the same time, essentially. And then what we used specifically was a model called BSLMM in the GEMMA program, which stands for Bayesian Sparse Linear Mixed Model. And what that one does, it is that it actually combines a ridge regression, which kind of makes an assumption that every variant has an effect, and then a Bayesian variable sparse regression, which makes an assumption that only a small proportion of the variants have an effect. This is good for when you don't really know what the genetic architecture of your trait is. So what you get out of this model is that it estimates the amount of phenotypic variance that's explained by both sparse and random effects in the model, and that is going to be referred to as PVE from here on out. The sparse effects are like your clearly detectable markers, like the things that you should be able to detect if you have the right condition for your GWA, and that's going to be referred to as PGE. The random effects are going to be like your infinitesimal effects of all of your markers, so the ones that have like effects that are so low that you can't really detect them easily. And then you can calculate like a marker set narrow sense heritability from these two values by multiplying them together. So what we're going to do is take a look at some of the results from me running this multi-marker GWA on our traits. And there are a lot of boxes here. Don't worry, we're only going to look at 3 of them. So the first one I want to look at is sex, which again is kind of acting as a pseudo control, and I'm using it here as sort of a model example of what high heritability looks like. So you've got 4 bars here. The first one is broad sense heritability, which I just put in there for reference. And then the second one is the narrow sense heritability that we calculated from our marker set. The third bar is the PVE value, which is the percent variation that's explained by our marker set. Variation in the trait that's explained by our marker set. And then the last bar is that PGE value, which is that portion of PVE that is supposed to be made of detectable loci that have a large loci, basically that have a large enough effect for you to be able to detect it. So what you see here is that it's just high across the board for these, but really the key is that for the PVE and the PGE value, the last 2 bars, is that most of the variation in our trait is being accounted for by our marker set, and most of that is detectable. And that comports with what we found in our GWA, because we actually did find a number of hits for sex that we already know are actually related to sex determination in aspen. So that's kind of what you would expect to see for a trait that would be kind of the ideal trait to run through a traditional single marker GWA if you are expecting to actually find the majority of the variation accounted for. So if we want to look at bud break, this one is more of an example of moderate heritability. So you've got a pretty high percent of the variation that's being explained by your marker set. I think it's really close to 70%. And of that, there's only really about like 25-ish% of that that's being explained by loci that are detectable. So that also kind of comports with what we found with our GWA, where we did find some hits for bud break, but not a ton. There's a couple of reasons why you may not see this trait as being more highly heritable in our particular situation. One being that I know we are missing some important genes for bud break, partially because of the way that our sequencing was done and the nature of the genome for Populus species having whole genome duplications in it, and having to not being able to keep some of those genes in there because you can't get like a probe to map to it specifically. So there, I know there's a little bit of an issue for this trait specifically because of that. But even still, we still did actually find some interesting candidate genes for that trait. So the last one I want to talk about is the phenolic glycosides. And here's an example where you have more of a low heritability trait, where there's still like a decent amount of your marker set that's explaining the variation in that trait. It's around 66%, I believe. But the amount of that that's actually detectable loci is only around like 10%. So most of what is contributing to the variation of this trait in our marker set is not detectable. So those loci have effects that are low enough that we can't really detect them. And another way to look at this is to just look at the marker set narrow sense heritabilities that we calculated. And this kind of gives you more of a sense of what most of the variation explained by our marker set means for detecting associated loci. So Across all traits, it's less than 20%, even for highly heritable traits like bud break. So even though our marker set may explain up to like 68% of the phenotypic variation for bud break, only about 26% of that's attributable to detectable loci. Jasmonic acid, so there's bud break. Jasmonic acid over here is another one that's kind of got a similar trend to bud break, and we did find a couple of candidate genes for that trait as well. And then back over here with phenolic glycosides, you've got 66% of the variation in the trait being associated, like, sorry. So our marker set is accounting for about 66% of the variation in that trait, but only 8%, basically only like 14% of that is detectable. So you've got like a heritability of like 8%, which is really, really low. Let's see. Okay, so what is the genetic architecture telling us? We're going to go back to this little figure here. This is another way to think about what I was just talking about. The traits of the lowest PGE values, like phenolic glycosides, have underlying loci that are falling into this hard to identify genetically category because their effect sizes are so low. And most of the associations we did find are ones that had more common allele frequencies. So they're down here, even though their effect sizes were still low to moderate. So the question that I thought about when I was looking at this is whether or not some of these traits like phenolic glycosides could be subject to polygenic adaptation, where you have like these small shifts in allele frequency of certain alleles at these multiple loci that are basically trying to drive that trait value towards an optimum. And that's a really hard thing to detect using the methods that we're looking at, that we used, because when you have something that's highly polygenic, where there's many genes affecting the trait, and not necessarily any of them with large effect, you've got a lot of different allelic combinations that could give you that optimal trait value. And those types of adaptive, like polygenic architectures, you're not necessarily going to expect to see any of those alleles go to fixation. And that's something that is often used as like a signature to look for when you're looking for genes that are involved in controlling a trait. So what did we get out of the GWAS analysis that we did? Well, we have polygenic traits. Most of our traits are polygenic, which is not that surprising because most of our traits are also quantitative. So there's a lot going on with them. There's a lot of underlying genes that are probably involved in their regulation and even more directly, making those compounds. And then you also have some of the environmental feedback that's playing a role in how things are expressed for those genes as well. And the architecture, kind of look at the architecture we got from the multi-SNP GWAS is telling us that there are probably a lot of loci with really subtle frequency shifts and low effects that could be behind traits that have low narrow sense heritabilities like PGs. And the other, I mentioned before, there was another study that had used this method to look at a tree species, and they looked at a hybrid aspen of tremula and alba. And they also did look at some phenolic glycoside compounds, and they found kind of the same genetic architecture pattern I did. And I think they used rad sequencing instead of capture-seq like we did. But yeah, very, very similar results there. Another thing that could be contributing is rare variants that could have moderate to large effects. That could also be contributing to us seeing kind of this low narrow sense heritability, because we know that rare variants can be important for forest trees. There was a pretty big study in Pinus Tata that was done that showed that. And then also, like, we know that we may not see these variants if our sample size is not big enough. Like I had mentioned, you really need at least 1,000 at minimum for that, for you to be able to detect really rare variants. And then lastly, I kind of alluded to this before, but we may actually be missing some important genes or genomic regions in our genomic data. We don't have every single gene covered. I mentioned before, we have about 20,000 genes that were covered. There's about 36,000 genes in the aspen genome. So we're missing some genes. So some of that could be there. The other thing too is that there is a known role for non-coding variants in gene regulation. And because we only sequence genes, we don't have any of that non-coding information at all in this. Now, I do think that if we were to, actually have all the genes represented, all the non-coding regions represented, we would probably find more, but we still would not account for most of the heritability we would expect to see. There would still be a lot of missing heritability. And the reason why I think this is because most other studies, genome by association analysis studies that have been done in trees that have had the resources to do whole genome and have larger sample sizes have not been finding that much more than we have. And I think that that points to the genetic architecture of the traits playing a pretty big role in our ability to detect variants that are contributing to the majority of the heritability of that trait. So that's something that we have to figure out. Well, how are we going to kind of get around this kind of limitation of genome-wide association analyses to be able to find some of those things that are low effect, low frequency. And there are a lot of different ways to do it, but because we were particularly interested in trying to generate some more candidate genes for phenolic glycosides, what I ended up doing is a differential expression using extreme phenotyping. And so on the same day, I went out to WSS and I collected foliar tissue from 30 high PEG genets with a range of 10 to 16% dry weight phenolic glycoside concentrations, and then 30 low PEG genets, which their range was somewhere between 1.3 and 2.5%. And to give you an idea of what that means, I mean, I know those numbers are pretty far from each other, but also an important piece of information to keep in mind is that PG concentrations have to be somewhere above 7% for them to have a meaningful effect on herbivores in terms of their survivorship and feeding. So the good thing about where our two groups are is that one of them is clearly under that and one of them is clearly well over that. So I took the samples that I collected and we had total RNA sequenced, and then I did a differential expression on that, and I identified 1,243 differentially expressed genes. I used an adjusted p-value cutoff of 0.05 and then a log 2 fold change cutoff of 0, which basically means if it was differentially expressed in the high PG group as compared to the low PG group, then I looked at it as long as the p-value was below our cutoff. And the reason why I felt comfortable doing that is because our sample sizes are so high. Most RNA-seq projects don't have sample sizes that high. They'll have like 2 or 3. There is also like some noise I'm going to end up having to address, which I will talk to you guys about that in a minute because of the way that I did my experimental design, but I don't think it really inhibited us finding too much. So I'm going to jump right into the results from that differential expression. This is a volcano plot. It's a good way to kind of get an overview of what your differential expression results look like. So on the x-axis, you have the log 2 fold change values, which is basically how much something was up or down regulated. On the y-axis, you have the adjusted p-values, and they've been negative log 10 transformed just for better visualization. And the dashed horizontal line is where my p-value cutoff is. So anything below that is not significant based off of my adjusted p-value, and anything above that is significant. Then I did not, like I said, I did not actually have a cutoff other than it just had to be above 0 or below 0. Above 0 would be upregulated, below 0 would be downregulated. So I just have these other two vertical dashed lines in there to kind of like show you that most of what I found that was differentially expressed at a significantly differentially expressed only had smaller fold changes, so somewhere around 1.5 to 3 times up or down regulation. So all the red dots here are essentially differentially expressed genes that I'm looking at. Okay, and that's the main thing that I wanted you guys to take away from, you all to take away from this, is just that the up or down regulation is somewhat subtle. Like there are some that are a little bit more out there than others. But here's a heat map of the top 30 differentially expressed genes by adjusted p-value. I want to first kind of zero in on this bar on the top. This is our PG group. So if a genet is part, each box is a genet. If it's part of the high PG group, it's orange. If it's part of the low, it's this pink color. And ideally what you want to see is complete separation of your groups, which we have pretty good separation. It's not complete. There are a few low ones that are kind of interspersed in this high PG group. But our experimental design isn't kind of like a typical one where you have like a control and a treatment and you know, have the same basically individuals in the control that you do in the treatment. So we don't have that. We have basically groups that are defined off of their PG concentrations. And there are other things that some of these trees might share in common. That's going to make that a little bit noisier. Plus the trait that we're looking at, the phenolic glycosides, is quantitative and it's controlled by a lot of genes, as we found from our multi-SNP GWA looking at the architecture. So we've got some noise in our data, and what this kind of looks like, to give you another picture, is this is just one gene, the count data for one gene, and it's the one with the lowest adjusted p-value, so the one that's at the very top of this heat map. So you've got your normalized counts, which is how many copies of the gene were being expressed essentially on the y-axis, and then you've got your two groups, your high PG group and your low PG group, and then each dot is a genet. So what this is kind of showing us is that there's a lot of variation in the counts for even this gene. For the high PG group in particular, it's a little bit more clustered for the low PG group, but even there's some variation, and there's definitely overlap between the two. So this is kind of another way to look at like why our data is a little bit messy. Our sample size is really high. Oh, sorry about that. So I'm not too concerned about that overlapping a little bit. So going back to the heat map, the main thing to note from this for at least these top 30 differentially expressed genes by adjusted p-value is that overall you've got genes that are downregulated in low PG genets as compared to the high PG genets. And most of the functions for these genes, there's a lot of them that are involved in catalytic and enzymatic reactions, a lot that are involved specifically in transferring acyl groups, and it could be involved in phenolic biosynthesis. So that's interesting, but what I would like to know is, out of this 1,243 genes that we have, how can I figure out how to kind of like group and categorize those to figure out which ones are worth looking at and which ones aren't? Because we do have some noise in our data. And one way to do that is to do a cluster analysis. We use soft clustering because it actually allows you to have genes be a member of more than one cluster, like hard clustering where it's either a member of one or it's not, essentially. So this is a way to do cluster analysis and then also kind of give a little bit for some of the noise that's in our data. So what you do is you cluster the genes together using this analysis. I used a program called mFuzz. And then what you can do once you've done that is you can take those clusters and do a Go enrichment analysis, gene enrichment analysis on it, and see what kinds of functions are enriched in those clusters of genes. And there were two major clusters of interest that I found. And the first one was a lignin biosynthesis and regulation cluster. There were 85 genes in it. This one's really interesting to me because there is some evidence that there are some growth defense lignin trade-offs, and there might be some co-regulation going on between both like defense biosynthesis pathways like phenolic glycosides and then also lignin biosynthesis. So I think that those are probably a good set of candidate genes to look into more. And then another cluster was for secondary metabolites. There's 123 genes in that cluster, and those were all upregulated in the high PEG group. And I think that those are probably also another group of good candidates for looking at genes that might be involved in phenolic glycoside biosynthesis. One note that I will make about a lot of what we found in both the GWAS and then also the differential expression is that a lot of the things we found were more regulatory, a lot of transcription factors too that don't just play roles in like, you know, defense regulation, but they play roles in all sorts of different things going on with an organism, growth and development. So there's probably a lot of crosstalk between how these different things are regulated, which is also probably adding another fun layer of complication to trying to figure out what's controlling some of these really complex traits. So just to summarize, the main things to take away from this is that growth and defense traits for Populus tremuloides are likely polygenic and have many low effect and rare alleles that are accounting for that trait variation. And then also that using other tools like differential expression can help you find an alternative way to generate candidate genes for traits that have that kind of architecture. And then I just wanted to say thank you to everyone who has worked on this project and made what I just presented on possible, including my funding sources and colleagues and mentors, and all the undergraduate students as well. And I can take any questions now.
Thank you, Jennifer.
Thank you.
If anybody has questions, feel free to just shout them out, or you can type them in the chat as well.
I have to figure out if I can see the chat while I'm sharing a screen. I might have to stop sharing for a minute. Oh wait, no. Here we go. Found it. I don't see anything in the chat right now.
Yeah, there's nothing in the chat.
Okay, just making sure.
Okay. Oh, so here's a question. Are there general lessons we can learn about trade-offs between defense and growth?
Yeah, so I didn't really go into this too much. There's been some other work that's been done in Arabidopsis that has looked at how they could actually decouple that trade-off between growth and defense and actually have the plant grow and defend like to the max. And they were kind of finding out that a lot of it is about how the pathways are regulated. So I think that's where a lot of the connections are going to end up being between these different allocation things that the plant has to allocate to, whether it's growing or defending, is going to be about how those pathways are regulated, and not necessarily like whether or not they have like enough resource to do both. That's also why I think it's interesting that a lot of what we found is in that realm of regulatory components, because especially for the differential expression, because I think that's probably where people are going to have to go to find those answers. Okay, let's see another one. Can you explain a little more about the difference between PVE and PGE? Yeah, so we're talking about like a model here, so it's not necessarily perfect. But what you're getting out of it is like when you do like a single marker GWA, for example, you can get like kind of like an effect size for like how much of the variation that particular like SNP is accounting for in your trait. So that's like the whole point of a GWA, right? Like you're taking variation in a trait and variation in the genome, and you're going, is there any places where those two things are associated with each other? And so like when you do a single marker GWA, what you do is you're only looking at one spot in the genome and then comparing that to that phenotypic variation and seeing if it's associated. And then you have to do that for every single marker. What the multi SNP marker does, well, actually, let me step back. So that means you can actually kind of get like a specific effect size for a specific marker when you do a single marker GWA. When you do these multi-marker ones, instead of looking at like one marker at a time, you're taking your entire like marker set and then saying how much of the variation in the trait is explained by this entire marker set. So I'm not going to get like specific like effect sizes for individual markers. I'm just going to get an overall number for that, and that's what the PVE value is. It's telling us how much of the variation in our trait is being explained by our entire marker set, like all of those SNPs together. And then the PGE value is basically a proportion of that PVE value. It's telling us the percentage of that variation that's explained by our marker set, how much of that is actually loci that probably have effect sizes that are large enough that we might actually be able to detect them, versus them just being kind of like so small that they're not really measurable, at least not in the way that we're trying to measure it. I hope that helps. Let me see. Have you thought about strategies to combine your GWA and DE data? Yes, I have. I just have not had time to do it yet. We are somewhat limited to like by the fact that I don't have RNA-seq data for all of the trees at WSASP. I only have it for 60 trees. So if I want to do like what you would call like an EQTL GWA, where you basically take that sequencing data, like that expression data, and treat it like it's a trait, and then run it against your genome, we can do that. But then the other limitation we have is that we don't actually have whole genome sequence data. So we only have the exome, and that's why I can't really do a whole lot with what I have. Like we would have to be able to invest more in basically getting like, say those 60 samples I have whole genome sequenced, and then we could probably think about doing something like an EQTL GWA.
That pretty much leads to the next question in the chat.
Oh, yeah. Oh, yeah. Yeah. I mean, I don't know right now what I know. Let's see. Yeah, I'm not sure what I'm going to do yet, and that kind of relates more to where I'm going right now with my life. So, and I'm not entirely sure whether or not Rick's lab has any plans to do much more. I know our collaborators in Sweden, we work with Pella Ingvarsson. He was at Umeå University, he's at Uppsala University now. I know they've been doing a lot of work with this. They're the reason why we had a reference genome of Populus tremula to align our stuff to, instead of having to use trichocarpa, which was nice because they're more closely related to each other, tremula and tremuloides are. And they're also working on like whole genome sequence, like reference genome for tremuloides itself as well. So I know that group is definitely going to continue doing more, and I'm hoping that I will be able to continue to do some more with some of this RNA-seq data that I have. So I don't know specifically what I'll do yet, but I would like to do some more. Okay, you mentioned how differential expression is an alternative way to identify candidate genes. How did you identify them initially? Well, so we were using genome-wide association analysis initially to identify candidate genes that were involved in the traits that we were interested in. And like I had mentioned in the talk, we just did not find that much, which is actually a common problem for forest trees. A lot of studies that have been doing GWAS in forest trees have just not been finding nearly as much as they expected to. And I think a lot of it is because most of the traits they're looking at are quantitative. And this is not necessarily limited to trees. Like if you go to human genetics and you look at what they're finding when they're actually looking at things like height or depression or diabetes or diseases that are actually very polygenically controlled, they're having the same problems we are. They just have slightly better resources than we do. so there's that too. Let's see. Possibility of assortment bias when doing, yeah, I mean, there's definitely a possibility of assortment bias happening. That's definitely an issue. And I think that's the thing, like, I mean, I know I focused a lot on the architecture of the traits because I do think that that's a big deal for this, but it doesn't mean that it's mutually exclusive for some of these other issues to be at play with what's happening. I just think that having a lot of genes that are likely affecting the trait variation is probably playing one of the bigger roles in why we're not finding as much. Let's see. Yeah, okay, so here's another question. My question is similar to Carlos's question. Do you think analyzing intron data would be valuable in your research? Yeah, I mean, I'm sure it would be useful to look at that, but once again, like, you know, part of it is just the limitations of what you can afford to do with, I mean, even what we did do, like that common garden got started before I even started working in that lab. It was there for almost 5 years when I started working in that lab, and it was just a massive amount of effort that they had to put together to put that garden together and then maintain it. And then it was a massive amount of effort for us to collect all that phenotypic information. And it was, you know, not necessarily as hard to get the material to be sequenced, but then just even the sequencing was it's not cheap. Like, I mean, I know it's getting cheaper, but it's not cheap, especially when you're doing like 500 individuals. So, yeah, okay. So thank you guys for your time and your awesome questions.